Hello everybody, welcome back to another episode of STEM Nerds. I'm Jason. Glad you're here today because in today's video, we need to talk about the adaptive immune system. In the last video, we talked about the innate immune system. If you haven't seen that, you need to watch that first. I'll put that video in the upper right-hand corner. But that video is really important for understanding this one. So adaptive immune system, it is what helps protect us long-term against infection. Let's take a closer look. So if you remember, we talked about the innate immune system, always on, always present, and non-specific. The adaptive side of our immune response is a little bit different. It is very specific, and it is generally only active when there is an infection. And the other thing is it provides memory for our immune system. We can remember these infections and then fight them a lot easier later on down the road. So if you remember in the last video, we talked about the innate immune system cells. Those were all the phils, basophils, eosinophils, neutrophils, and then we also talked about the monocytes and the macrophages and the dendritic cells. Well, in today's video, our focus is going to be on T cells, B cells, and natural killer cells, but we're still going to need those cells from the innate immune system to help us out here. All right, so before we get into the cells, we need to talk about the lymphatic system. It is hugely important to our immune system and to our overall health. A lot of people think when you drink water, it goes into your esophagus, stomach, small intestine, large intestine, and somehow out. Well, how does it wind up in the bladder? Well, when you drink water, what's actually happening is it goes into your esophagus, your stomach, your small intestine, and in your large intestine where it gets absorbed into the body. Once in our body, it gets into our circulatory system, eventually into our cells and into our interstitial spaces. Well, we have to be able to reclaim that water, and that is the job of the lymphatic system. Its primary role is to reclaim that excess water. And then eventually it returns it back into the circulatory system where it gets filtered out by the kidneys, winds up in our bladder, and then we eventually excrete it as waste. Now, it's important to note that the lymphatic system doesn't have a little lymphatic heart, and so it's not pumping through our body in the same way that our blood is. In fact, lymphatic fluid moves best when somebody is active through muscle contraction and movement. That's why people will often say, usually you're going to be healthier if you are active and moving around. Associated with the lymphatic system, we have what are known as lymphoid organs, and we have primary and secondary lymphoid organs. The primary lymphoid organs are your bone marrow and the thymus. And the bone marrow and thymus are primary lymphoid organs because that is where the immune system cells originate. B cells mature in the bone marrow and T cells mature in the thymus. The secondary lymphoid organs are often areas where antigen is encountered, where the bad guys are encountered. And so that would be things like the lymph nodes, adenoids, tonsils, mucosal-associated lymphoid tissue, otherwise known as MALT, skin-associated lymphoid tissue, otherwise known as SALT, Peyer's patches. You also have the spleen and the appendix that are associated as secondary lymphoid organs. So I just said that the secondary lymphoid organs are often where antigen is encountered. We do need to take a couple seconds and talk about, well, what is an antigen? An antigen is an antibody generator. So it's something that gets into the body and elicits a secondary immune response. In other words, you're going to get activation of T cells and B cells and eventually antibodies. Examples would be viruses, bacteria, protozoans, fungi, parasitic worms. Sometimes, incorrectly, you'll have environmental antigens like dander and dust and pollen that can elicit that immune response as well. There are a lot, thousands upon thousands of different types of antigens. All right, so how do we get the adaptive immune system cells involved? Well, in the previous video, we talked about the importance of the innate immune system. And at that time, I said that there were two cells in particular that were going to be important for getting the adaptive immune system involved in an immune response. They were antigen-presenting cells. It was the macrophage and the dendritic cell. Now, this picture is really important. It's illustrating just how the antigen-presenting cells, macrophages and dendritic cells, allow the adaptive immune system to get involved in a fight. So what we have here on the left-hand side is we have an antigen-presenting cell that's kind of the yellowish-looking cell. And what happened is that antigen-presenting cell, if you remember from the last video, it took that cell, took that pathogen in on purpose. Important term for us to understand is when an antigen presenting cell takes in a virus or bacteria on purpose, we say that is exogenous antigen. Exogenous antigen is taken in by an antigen presenting cell, chopped up and presented to the adaptive immune system. Now macrophages, as we're seeing here on the left-hand side, macrophages will present the antigen that they've found, that exogenous antigen, to a T helper cell. Importantly, the way that the macrophage presents that antigen is on something called MHC class II molecules. So antigen presenting cells have MHC class II molecules. It's kind of like their arm. The antigen is sticking out and it's presenting it to a T helper cell. A T helper cell is going to have a very specific region, a variable region on the end of its receptor that recognizes the antigen that's being displayed, in this case by the macrophage. As a result of this interaction, a couple things are going to happen. The T helper cell, its role, its job, is kind of like the middle manager of the immune system. It's an immune system coordinator. What it's going to do is it's going to tell the macrophage, hey, you know what? This is actually something bad because my receptor has recognized the antigen that you've displayed. And so it's going to provide some cytokines to the macrophage to make it active. And so what happens is that macrophage actually becomes something called a giant cell. It goes back to the site of the infection. It's going to be physically larger, better at opsonization. We defined that term in the last video. That means better at recognizing pathogens and gobbling them up. And they're going to reproduce in higher numbers. At the same time, the macrophage is providing cytokines that are telling the T helper cells grow and divide because there's something in the body that needs to be fought, and we need a lot more T helper cells. We need a lot more coordination and a lot more, at this point, pro-inflammatory cytokines flying around the body. As a result of the T helper cells becoming more numerous, they're also going to encourage the growth of cytotoxic T cells. And you can see here on the far right-hand side, we're getting the activation of our cytotoxic T cells. And so our T helper cells are the middle managers. Well, what do the cytotoxic T cells do? Cytotoxic T cells also have a very specific receptor that would be similar, if not exactly the same, as the helper T cell. But the cytotoxic T cell has a little bit of a different job. It is a warrior. It's a soldier. And what it's going to do is it's going to go to the site of the infection using that very specific receptor, and it's looking for self cells, non-immune system cells that are sick, that are displaying that same antigen, but they're going to do it a little bit differently. The cells that are sick are going to display antigen on MHC class I molecules. These are major histocompatibility molecules. That MHC class I is presented by all cells in the body. And what it's really doing, the purpose is an MHC class I, when there is a protein on the end of it, it's telling the immune system I'm either healthy or I'm sick. If it's displaying abnormal antigen, it's telling the immune system I'm sick. If it's displaying normal antigen or a normal protein on MHC class I, the immune system generally just leaves it alone. If it's not displaying anything on MHC class I, that can also be a problem that would elicit an immune response. So if an infected self cell is displaying antigen via MHC class I, we say that that antigen is endogenous. Endogenous antigen means that the cell was actually infected. It's reproducing that antigen, it's displaying it, it needs help in the form of that cytotoxic T cell. The cytotoxic T cell, what it's going to do when it recognizes the antigen is release something like a tumor necrosis factor or some type of cytokine that is going to tell the self cell that is sick to undergo apoptosis. Again, we've talked about this a couple times now. Just like with interferon, when interferon told the cell, hey, you know what, you need to get rid of yourself to stop spreading virus, the same thing is true here. If there is an intracellular parasite, the cytotoxic T cell can encourage the self cell that's infected to kill itself off so that the infection doesn't spread as quickly. Now, there's one thing I want to mention here, and it's really important. We kind of focused on the macrophage at this point, but there's another antigen-presenting cell, and that is the dendritic cell. Remember, that was the one with the little arms, kind of looked like an octopus. The dendritic cell has a little bit of a special ability. Just like the macrophage, it can activate a helper T cell via MHC class 2, but dendritic cells also have MHC class 1, and they can encourage the growth and the differentiation of cytotoxic T cells. And so it can actually encourage the division and activation of both types of T cells. Now, it is important to note that this entire process at this point is happening in a secondary lymphoid organ. And so if you think about the nature of an infection, let's say somebody is infected with a respiratory virus. We said last week, or in the last video, that neutrophils were generally going to be the first on the scene, followed very closely by macrophages. Their first attempt is to clear the infection. If the infection can't be cleared, they're going to try to wall it off and try to surround it so that it can't spread. But in the meantime, if that infection does start to spread, you have the macrophages and dendritic cells bringing the exogenous antigen to a secondary lymphoid organ, and that's going to help get the activation of the T cells, both the helper T cells and the cytotoxic T cells. And so we're really beginning that adaptive immune system response. So I want to pause here for a second because we've said a few things that really need some explaining. Earlier, I mentioned that antigens can number in the thousands, tens of thousands. There are so many different potential types of antigens, things that would elicit an immune response, especially an antibody response. The problem is our genome only has about 20 to 21,000 different genes, and the vast majority are not dedicated to the immune response. It turns out that there are segments of our genome that are dedicated to that response. And if you mix and match them almost like a buffet, you can get a lot of different variable regions that would allow T cells and B cells that we're going to talk about in a few minutes and antibodies to recognize almost an infinite number of antigens. Well, how does this happen? I like to refer to it as VDJ. What that means is there's a segment of genetic material dedicated to producing regions, variable regions of receptors and antibodies. And there are about 40 or so different variable regions that can be chosen. There's about 20 to 30 different diversity regions, and there's a few different joining regions. But when you combine all these different options together, and this can often happen randomly, you wind up with 10 to the 16 different combinations. In other words, a 1 with 16 zeros after it, and so you're pretty much covered with an almost infinite number of possibilities. Thus, the reason why the immune system can recognize eventually just about any type of antigen that would ever exist. Now, that does create a bit of an issue. If these variable diversity and joining regions are kind of being mixed and matched somewhat randomly, you could imagine a situation where you get a variable region that actually recognizes self as a foreign entity, and that would be horrible. We call that an autoimmune disease. And so generally speaking, what happens if the immune system is functioning properly, any T cell or B cell that has a receptor that recognizes self protein is killed off before it ever leaves its site of maturation, be it the bone marrow or the thymus. And so because we've got all these different regions that can be mixed and matched together, we have to make sure that the body is not creating T cells and B cells that can target self as a potential antigen. All right, so now let's take this a step further. Let's go back to our respiratory infection example. We have our macrophage or our dendritic cell, and you see that in the upper left-hand corner. But in this case, we'll call it a macrophage because, well, that's how the picture is labeled. And we see the macrophage is displaying what it's found. Remember, that was exogenous antigen. It's displaying it via MHC class 2 because that's what the antigen-presenting cells do. It's not sick. It brought it in on purpose, chopped it up, and is now showing it to the T helper cell. That T helper cell has a very specific receptor for that particular antigen. It becomes activated. Now here, we need to get the B cells involved. The way the B cells get involved, believe it or not, they too are antigen-presenting cells, just like the macrophage, just like the dendritic cell. And so they take in, using a very specific receptor, they will take in an antigen, chop it up, and display it via MHC class 2, just like macrophages and just like dendritic cells. That activated helper T cell has already seen this antigen. The macrophage or the dendritic cell showed it off earlier. And so now we've got more helper T cells, and so the chances of a helper T cell with that specific receptor recognizing the antigen that's being displayed via MHC class II from the B cell is much higher. What's going to happen as a result? The T cell is going to provide pro-growth cytokines to the B cell, tell the B cell, hey, we need you to grow and divide. You've got something bad. I saw this earlier from a macrophage or dendritic cell. Keep in mind, this is happening in a secondary lymphoid organ. So if the B cell is recognizing it there, chances are this infection has spread from the original site of where it entered the body. Now, the B cells are going to do two things. One, they're going to produce what are known as plasma cells and eventually produce memory cells. And that's part of where we get the immunological memory. These plasma cells are massively important because what they're going to do is eventually produce antibodies. Throughout this entire process, if someone was infected with, let's say, this respiratory virus, we really would have a hard time producing enough neutrophils, macrophages, T helper cells, cytotoxic T cells to destroy the virus. Because remember, viruses, one virus enters, there could be hundreds, if not thousands of viral particles leaving a single cell. And so we'd very likely lose that war. But when our plasma cells start producing antibodies, antibodies are proteins. And we can produce a lot more antibodies than viruses can replicate. And so these antibodies are hugely important to our secondary immune response and to the overall immune response. All right, so I think it's really important to talk about the general structure of an antibody. Again, it is a protein. I mentioned they have this little variable region. And so there are some things I think we should know about antibodies. Thing number one is you'll see the very, very end, it's shaped like a letter Y. The very ends of that letter Y, in the top left and top right, those are listed as hypervariable or variable regions. I just call them the variable region. This is the region that was created by that VDJ, the variable and diversity regions. This is also the area where the antibody is going to attach to an antigen. Now, generally speaking, the variable region attaches to what is known as a specific epitope. So you might have an antigen, let's say it's an E. coli cell or an influenza virus. But those influenza viruses or those E. coli cells, they're really complicated. They have lots of different proteins, lots of different lipids sticking out. That variable region is going to recognize one specific type of protein, and that specific region where they bind is called the epitope. And you could imagine then that you might actually have multiple helper T cells, multiple antibodies that bind to different epitopes on the same antigen. And so we have the variable region there. And then we have this FC region, otherwise known as the constant region. This generally cannot change because this immune system cells, as we're going to see in a few minutes, that need to recognize antibody bound to viruses and bacteria or antigen in general, they need that constant region to be constant. They have to be able to recognize it, so that cannot change. You'll also notice that we have a light chain and a heavy chain. The heavy chain is basically from where it's labeled hypervariable on that inner part of the Y, all the way down to the dark purple labeled FC. And so it's that entire area. The light chain is just from the hypervariable region on the outside where it's down to the purple right there. So you've got heavy chain, light chain. You have our variable region. We have our constant region of an antibody. Now, there are different classes of antibodies, and these are the 5 main types, and I want to make sure that we go over what these are and what they do. And so IgG, it is a monomer. It's a single antibody unit. You can see here, this is a lot easier to see the heavy chain and light chain. So an IgG in the upper left-hand corner, the blue is the heavy chain, and the purples are the light chains. And so IgG is hugely important. It's the most predominant antibody you're going to find in a body, and it is the one that is produced in high concentrations in both a primary and especially a secondary immune response. IgM is a big pentamer antibody. You can see here you got 5 antibodies stuck together. So it's a big structure, and because it's so big, it really doesn't get through epithelial and endothelial tissue well, and so it's primarily going to be located in the circulatory system or the lymphatic system, so not usually in the tissues. It's also the first antibody that's produced, and it's really good at what's known as agglutination. I want you to think of agglutination. It's got the word glue in it. And so agglutination is basically the ability of antibodies to stick to antigens and then stick them all together. And we're going to explain what that means and why it's so important a little bit more in the next slide. IgA is a dimer, so you've got two antibodies stuck together, and that one is important because that is the antibody of secretion. Generally speaking, you're going to find IgA in mucous membranes, especially important in breast milk and saliva. And so if there is going to be antibodies technically outside the body, a lot of that is often IgA. IgE, that one for both IgE and IgD, their importance in an immune response is greatly diminished compared to the first three. And so IgE is primarily going to be more elevated when there are parasitic worm infections. It's also been linked to allergies, and it is an antibody that mast cells use when they're releasing histamine, and that's that allergy connection. So, but very small concentrations. And IgD is in such low concentration in the human body that scientists are really still trying to figure out what its primary role is, and whether or not it's even necessary, because there have been studies that have shown in genetically engineered animals, non-humans, that when you eliminate their ability to produce IgD, it doesn't have a massive impact on the immune response. So these antibodies are really important. In fact, they have 6 protective outcomes, and they're listed here. Antibodies can do immobilization. And so when antibodies bind to things like flagella, they can stop motile bacteria, which can also help prevent them from attaching. Antibodies are involved in neutralization. They can bind to viruses and all kinds of antigens and pathogens and prevent them from attaching. And we've said so many times that attachment is the first thing that a pathogen must do in order to cause an infection. Next is agglutination. We kind of hinted at this with the IgM in the previous slide. Again, agglutination is the ability of the antibodies to stick a bunch of antigens together. And so the way I like to explain it is, imagine you're sitting in a room with a dozen friends, and I open the door to that room and I take a 55-gallon drum and I just chuck M&Ms all over the room. It's a very large room, and your job is you need to clear up all these M&Ms because it turns out they're really dangerous. Now, you could go through and pick them up one by one, and that would literally take you forever. Or what if I gave you something that could agglutinate the M&Ms? And so when I chuck that 55-gallon drum of M&Ms into the room with you and your dozen buddies, you've got this substance that can stick them together into big, giant M&M balls. And now instead of trying to clear up thousands upon thousands of individual little M&Ms, you can maybe clean up a few dozen giant M&M balls. And I know that sounds kind of crazy, and some of you are like, wow, it's making me hungry. But that's what agglutination does. That's what these antibodies do, especially IgM. And it makes it a lot easier for the macrophages and the neutrophils and the dendritic cells and these cells that are responsible for gobbling up the bad guys. If they can find a clump of bad guys, antigen-antibody complexes, that's a lot easier than finding them individually. Next, we have complement activation, something we discussed in the last video. Complement is that protein that's involved in opsonization, inflammation, and cell lysis. And so when antibodies are bound, that can activate complement. And then I just mentioned opsonization. We talked about that in a previous video. Opsonization is basically the antibodies making it a lot easier because of those constant regions sticking out whenever something is bound by antibodies. So if you had a bacterial cell or a virus and it's got antibodies bound to it, the little y, that's going to be bound to the actual antigen, and that little tail is going to be sticking up. And that constant region allows the macrophage or the dendritic cell or the neutrophil to bind to that clump, take it in, and break it down. That's opsonization. The last one is called antibody-dependent cellular cytotoxicity, sometimes known as ADCC. This involves natural killer cells, and that's all I want to say about it right now. But when you see antibody-dependent cellular cytotoxicity, I want you to think natural killer cells, and we're going to talk about those in a minute. So as part of the immune response, as it grows, as our immune system is fighting a pathogen, a few things are going to happen. Thing number one is the immune system, especially B cells, are going to go through something called class switching. And what that means is they're going to switch the class of antibodies that are being produced. Remember, I said IgM is usually the first antibody that is produced, and then IgG is produced after that. Well, we're switching. The B cells are switching the class of antibody. That's called class switching. The other thing, hugely important, is affinity maturation. As the immune response matures for a specific type of infection, whether it's an influenza virus or a bacterial infection, as that infection progresses, the immune system gets better and better and better and more specific at recognizing that particular antigen and recognizing the epitopes that are going to be easiest to bind. And so over time, I mentioned already, but let's say you have an influenza virus and there are half a dozen different binding sites, and there are different immune system cells, different T cell and different B cell lineages, all producing antibodies that recognize those 6 different epitopes. Well, most likely, there's going to be 1 or 2 epitopes on that influenza virus that is going to be the one where the antibody recognizes it the best, it's the easiest to access, and it binds the tightest. When that happens, those antibodies that bind the strongest to an epitope, those are going to be the ones that are going to be produced more often, and those cells are going to be produced more often. And the last thing, of course, is something we've already talked about, and that is the production of memory cells, B cells. There are memory B cells. There are also memory T cells. And so both of those are going to be around so that if we ever encounter that same antigen again, we might not even know it because we'll have the cells there ready to fight this battle. Now, I mentioned natural killer cells before and how they were involved in antibody-dependent cellular cytotoxicity. So the natural killer cells, they're kind of interesting because they don't necessarily recognize specific antigens. However, we include them with the adaptive immune system because we need the antibodies, generally speaking, to activate the natural killer cells. And so what happens is you've got 2 cells in the adaptive response that recognize self cells that are infected and kill them off. But they go about things differently. So we talked about the cytotoxic T cell and how it would recognize sick self cells displaying antigen, endogenous antigen via MHC class 1. Cytotoxic T cell is a specific receptor. If it binds, it kills that cell off, prevents the virus from spreading. Natural killer cells are a little bit different because what they're doing is they're actually recognizing the constant regions of antibodies bound to antigen that are either being displayed by self cells or antigen that has been bound by antibodies that is a foreign entity like, let's say, a bacterial cell. They also have pro-apoptotic cytokines that will encourage cell death. So 2 cells doing the same job, they're just going about it a little bit differently. Now, I mentioned the concept of a primary and secondary immune response and how those memory cells really help us when it comes to the secondary immune response. And so this is a pretty decent example of what happens when the immune system encounters an antigen twice. And so in the first example, we have the primary immune response. And we're going to see that when we encounter an antigen for the first time, it's going to take time for the immune system the innate immune system to get up and running, to eventually alert the adaptive immune system to get enough cells there to produce enough antibodies to clear the infection. And so what we see is we have a period of time, and it's not a super strong response. You're going to get both IgG and IgM in that response, and possibly the other antibodies as well, depending upon the infection. And then hopefully the infection clears. And really important here, a cell that we haven't talked about yet, there will be T regulatory cells, yet another type of T cell that will produce anti-inflammatory interleukins that tells the immune system, hey, chill out, the war is over. Now, let's say we fast forward a few weeks, a few months, and we encounter that exact same antigen again. If we encounter that exact same antigen again, you're going to see in that secondary immune response, that purple line, we get an extremely fast response. And so in a secondary response, it's much, much faster because the memory cells are there and way stronger. And that's because those memory cells immediately start kicking out a lot of IgG. And so the primary thing that you need to know between primary and secondary immune response is the secondary immune response is way stronger, way faster. Please do not get this confused because a lot of people think the primary immune response is the innate response and the secondary is adaptive. That's not true. We're explaining what's going on with the adaptive immune response in both cases. All right, everyone, so that was a brief overview of the adaptive immune response. Again, if you haven't seen the innate response, I would highly recommend to do that. I'll put that video in the upper right-hand corner. As always, thank you so much for being here. Have a great week.